put a new unit 9.6 which is called as genetic marker. Small unit which we can understand easily if we understand what is genetic marker. So, first I will tell the title which is called as genetic marker. Genetic marker means what? Genetic marker means, as I told you in the beginning, on every chromosome there is a gene and that gene will be having a function. So, there is a chromosome, on the chromosome is because of DNA, the combination of DNA. In that DNA there is a structural part, in that DNA there is one particular sequence of nitrogenous bases which are like ATCG, that particular part will be having a function. That functional element is called as gene. Again I told you, gene, for every gene there will be a function, but that functions will be multiple, like for example, one gene is responsible for skin color. So, which skin color? It can be black, it can be white, it can be brown. So, that particular form of a gene is called as allele. What is coming out? What is the form which is out is called as allele. So, every gene will have function, multiple functions, that multiple functions are called as alleles. Now, coming to genetic marker. What is genetic marker? Genetic marker is also the same. So, on the chromosome there will be a gene, because of gene there will be a function. On a chromosome there is a gene, on the gene there will be a function, that resultant function which is coming out from that gene is called as genetic marker. So, the mark which is coming out because of gene, for example, chromosome number 9, on the chromosome number 9 there is a gene and that gene will have a function. That function is called as which blood group? Either blood group A or blood group B or blood group AB or blood group O. So, the resultant function which is coming out because of gene is called as genetic marker. For example, ABO blood group system. In ABO blood group system we have 4 blood groups, A, B, AB, O. So, that blood group A, in your blood the blood group A, in your blood the blood group V, in your blood the blood group AB, in your blood the blood group O is mainly because of a gene which is sitting on the chromosome number. So, gene will have a function, that function is called as, now there will be an antigen A, so blood group A, there will be antigen B, so blood group. Antigen is called as what? Protein. We discussed the protein synthesis. What is protein synthesis? In the nucleus there will be a DNA. What is present in the DNA will be shifted to mRNA and that mRNA will be coming out from the nucleus. In the cell there will be a ribosome. In the ribosome already tRNA will be there. This mRNA will come near the tRNA, mRNA will be matching. When tRNA mRNA matching is happening, amino acid sequence will be formed. That amino acid sequence is called as protein. What is example of protein? It can be a hormone, it can be a lipid, it can be a blood, it can be an antigen also. What is antigen? That is called as blood. So, a antigen will be formed in the form of protein. What is the antigen? That is called as blood. If antigen A, then blood group A. If antigen B, blood group B. What is antigen? Antigen is nothing but a protein. So, that protein antigen is because of what? In the DNA, the information which is present is translated into mRNA and the information now shifted to mRNA which is called as messenger RNA. Now, messenger RNA will be going for batch pairing or match pairing. In the match pairing in the ribosomes, it will go near the RNA and it will search for relevant tRNA, where there is mRNA, tRNA relevant matching, hence a amino acid sequence will be formed. That amino acid sequence is called as protein, which is an example of antigen. So, what amino acid sequence is formed? If A is formed, then it is called as A antigen. A antigen is, then it is called as A blood group. If B antigen is formed, then it is called as B blood group. If ABA antigen is formed, then the blood group is called as AB. Means if 2 antigens are produced, the blood group will be AB. If no antigen is produced, then the blood group will be O. So, based upon the protein which is formed, Protein is nothing but antigen A or antigen B or antigen A and B or no antigen. If A antigen is produced, then the blood group will be A. If B antigen is produced, then the blood group will be B. Means I want to tell you that in our blood there are 23 chromosomal pair, as you know. On that 23 chromosomal pair, on a chromosome there will be a gene. That gene will be responsible for a function. That gene will be responsible for a function. That functional segment, like a gene which is responsible for manufacturing of a protein which is called as antigen A, then the blood group will be. If a gene is responsible for manufacturing of an antigen B, antigen is called as protein. If a gene is responsible for manufacturing of antigen B, then the blood group will be called as B blood group. If a gene on the chromosome is responsible for manufacturing of antigen called as A and B, then the blood group will be called as AB. So, in simple words, blood is nothing but protein. Protein is nothing but antigen. Antigens are so many varieties like A, B, O, AB. That antigens are because of the information which is present in the gene. So, gene is responsible for manufacturing of a function. Gene is responsible for manufacturing of a product. Gene is responsible for the manufacturing of a protein. Gene is responsible for manufacturing of an antigen, and that antigen is nothing but blood group. So, the genetic marker, that gene which is responsible for a mark, my skin color is a genetic marker. The skin color is because of a gene. My hair color is a marker. This is because of what? A gene. The resultant outcome which is coming out, because of a gene is called as genetic marker. So, A blood group is a genetic marker, B blood group is a genetic marker, AB blood group is a genetic marker, O blood group is a genetic marker because of a gene. Gene is responsible for how genes are just go for protein synthesis in the nucleus, chromosome. On the chromosome there is a DNA. The information which is present in the DNA will be shifted to mRNA, and mRNA will be coming out from the nucleus into the ribosome. In the ribosome already tRNA is present, which is called as transfer RNA. So, the transfer RNA is fixed. It will be waiting for relevant information. The relevant information is mRNA. That mRNA is because of DNA. So, DNA will tell that go and manufacture antigen A. DNA will go and manufacture antigen B. So, the information which is present in the DNA will be shifted to mRNA, mRNA will go into the ribosome, in ribosome tRNA will be waiting. So, the mRNA will come with, we have to manufacture antigen A, it will go and base pair with tRNA. Hence, a amino acid sequence will be found which is called as antigen. So, antigen A is been made. That antigen A is nothing but a blood group. So, the blood group system which is present in your blood is because of antigens. Antigens means what? Proteins. The proteins are manufactured because of what? The information which is present in the DNA. So, that DNA will be having a limited code which is called as gene, which is a functional element. For any protein to be manufactured, a gene will tell that manufacture this protein, a gene will tell that manufacture this protein, a gene will tell that manufacture this protein. Accordingly, protein will be produced. So, that particular outcome which is coming out because of a gene is called as genetic marker. How many marks you are getting? It is because of how you are writing exam. How you are writing exam depend upon yourself, your mind. So, what is your mind? That will be your marks. What is your mind? That will be your marks. What will be the outcome? Will be because of gene. The outcome ABO blood group will be because of a gene which is present on the chromosome. So, the outcome which is coming out is called as genetic marker because of a, this is very, very important. The outcome which is coming out because of a gene is called as genetic marker. The genetic marker is because of the information which is present in the gene. So, this resultant is because of, sometimes we become angry on some people. When we become angry on few people, when we angry on some person, when they become angry, why they are becoming angry? Because of something happened in the history, because of something happened on the history. So, that history is called as gene and the resultant is called as, so the outcome which is coming out is called as marker because of what? The gene. In my blood, whatever may be the blood group, if my blood group is A, that blood group A is nothing but antigen A. Antigen means what? A protein. What protein? Protein A is manufactured, which is technically called as what? Antigen A. That antigen A only why it formed? Because my gene told. In your blood group, why blood group B? Because there is antigen B in your blood group. Why only antigen B? Your gene told to manufacture antigen B. So, that blood group is a genetic marker for your gene. Then why not AB? Your gene didn't told. Your gene told me to manufacture only B group, so I am producing antigen B. So, the resultant outcome which is coming out, how sir? Protein synthesis. Gene is because of DNA. So, DNA information will go into the mRNA, mRNA and tRNA match pairing will be happening. When the proper base pairing is happening, the output will come. So, what blood group antigen A should form? So, DNA will tell, hey go, I am giving information, mRNA go and manufacture A antigen. So, accordingly A antigen has been manufactured. In that way, the information here, the output here, the output is called as a trait or a trait, a behavior, a output, a outcome is called as trait, a characteristic. Skin color is a trait. A trait can be used as a genetic marker. ABO is what? A trait, a output. It is an output. A blood group is an output. A trait can be used as a genetic marker in the study of individuals, families or communities, provided the trait is genetically determined. A is determined by gene, B determined by gene. The trait should be determined how? Genetically. That trait will be called as genetic marker. Genetically determined and has a simple pattern of inheritance. What is the simple pattern of inheritance? It will tell what the gene information, that what is coming from father, what is coming from mother, that will be in the child. Accordingly the gene will be there, accordingly the output will be there. For example, my father is A, my mother is B. Obviously my blood group will be A or cannot be none. Why? Because either A will be there or B will be there. Why? Because I pumped A and B from my parents. So it is genetically determined or not? Simple pattern of inheritance or not? So genetical marker is also determined by simple pattern of, it can be A or it can be AB or it can be AB, but it cannot be C, it cannot be D. Why? Because I got it from either from father or mother. What does that meaning? Simple pattern of, simple understanding. Oh, coming from father, coming from mother, and can be classified accurately and has variations common enough to permit to be labeled as genetic polymorphism. Again, in a classroom, my blood group is A, his blood group is B, her blood group is AB, her blood group is O, his blood group is O, his blood group, why differences? Genetic polymorphism. No two individuals will have same gene. How I can tell? No two individuals in a classroom are, or all the individuals in the classroom are not having same blood group. Every individual is having a different blood groups. Means what? The best example is genetic polymorphism through blood groups. It should be genetically determined and it should be an example for or. Genetic marker is a gene or DNA sequence determined with location, with a known location on chromosome that can be used for a specific function of an individual. That can be a specific function of an, that is called as, so the gene which is responsible for specific function. For example, likewise so many are there. For example, RH, rhesus, a protein which will be sitting on lymphocytes, which will be sitting, rhesus protein. How I got rhesus protein? Rhesus is a protein which was been firstly found in monkeys, then it is also present in human beings. There is one protein is there, actually it does not have any function, but it is there in my body. RH, rhesus protein. My gene told go and manufacture rhesus protein, so rhesus protein has been manufactured. It is present in my blood, it will be sitting on WBCs. So if I have rhesus, it is called as A positive. If I have rhesus on B blood group, then B positive. If I am having rhesus protein on AB, then it is called as AB positive. If I am having rhesus on O, then it is called as O positive. So presence of that rhesus protein is positive. Why this rhesus protein? Because of gene, no? Gene is because of DNA sequence. The information in the DNA sequence pumped into mRNA, mRNA coming out, in ribosomes tRNA is there, mRNA, tRNA, if information is matched, then rhesus has been protein. So presence of rhesus is called as Rh positive, absence of rhesus is called as Rh negative. But the existence of Rh is because of, gene. If gene tell, then Rh will be there. If gene doesn't tell, there will no. Means what? That genetic marker rhesus which is coming out is because of gene. If gene there, then rhesus protein is there. If gene is not there, then there is no. It can be A blood group, B blood group, AB blood group, O blood group, rhesus protein. Any outcome is because of gene. If gene is there, then there is outcome. Outcome can be anything. Several anthropological studies based on genetic markers were carried out to understand the genetic variations among the indigenous populations, mainly to study regional, ethnic and religious groups, to understand the genetic history and affinities of the population. So, here we are understanding genetic marker not from biology point of view. Here we are not understanding genetic markers from the point of view of biotechnology. We are understanding genetic markers from the perspective of population. For example, which blood group is present in South India, which blood group is present in North India, which blood group is present in Mongoloid people, which blood group is present in Caucasoid people, which blood group is present in Negroid people. In that way, we will be understanding the population variations by using the studies called as genetic marker. Population variations we will be understanding. For example, I will tell you, in a population, for example, in a population, rhesus proteins are present more in males, rhesus proteins are not present in females. Rhesus protein is present more in males, rhesus protein is not present in males, females, not present in females. Two conditions I am telling. Rhesus protein is present in males, rhesus protein is not present in females. Now both got married. So mother is pregnant now. So mother, I told, what is the condition? The rhesus protein generally not present in female. So there is higher probability that the mother doesn't have rhesus protein. So there is a chance that from her it can pump into the baby, so baby can also not have rhesus. So mother Rh negative, baby Rh negative, no problem. But more number of males are having what? Rh positive. So there is a probability that rhesus can come from father. Rhesus come from father. So if the baby is having rhesus which is taken from father. Now the baby is having Rh positive and the mother is Rh negative. So this Rh negative body will think that what is this new protein came, what is this which is not there in my body, there is some foreign particle came and the antibodies of mother will attack Rh positive. It will go and attack the protein, it will go and attack the rhesus protein. So in the process of attacking the rhesus protein, it will also destroy all the remaining cells. So the baby will be, the second baby will be died when there is Rh incompatibility. So what conclusion I came? In a population where the rhesus proteins are more in males and rhesus proteins are less in female, there is a chance of Rh incompatibility, there is a chance of disease called as erythroblastosis. So if I understand, then I will be alert. In my population Rh negative is less, females are having Rh negative, then what I should do? So every pregnancy is a doubt, every pregnancy is a doubt. For every pregnancy we have to go for a check whether the baby is having Rh or negative. If the baby is having Rh positive means concern. If the baby is having Rh positive means concern. Why? Because mother doesn't have Rh. So what we have to do? So they have to put investment on research and development. They have to put money on what? Pharmacy. They have to put amount on what? Injection, manufacturing something, or they have to put money on artificial proteins. So they have to artificially imbibe protein into the baby, so that baby is Rh positive should be neutralized, so that Rh negative of the mother or the antibody of the mother will not attack the Rh positive of the child. That variations we will be studying in anthropology. In anthropology we will understand what is genetic marker. In anthropology we will understand what is blood bank, and we will apply this in the community. So anthropologist will be worried when the Rh negative is more in population in female. Then he will tell to the community people or the leaders that there is some emergency will be there. There will be more number of erythroblastosis, footalysis disease will be there in your community. Be alert. So they will be alert. In that way, we will be using the knowledge of anthropology in solving the medical issues of a community. So first I will take the blood group example, ABO blood group. The genetic marker like blood groups, blood enzymes, human leukocyte antigen and serum proteins studied extensively to understand inheritance. These all are proteins only. blood is a protein, enzyme is a protein, serum is a protein, egg is a protein, sperm is a protein, ova is a protein. These all are what? These all are proteins. Proteins are because of what? DNA, the information which is coming out from the DNA according to the gene. What is the formula? According to the accordingly outcome. So, first we will discuss ABO blood group system. So, blood group A, then what is visible? A. Phenotype will be called as A. Antigen will be called as A. Because of antigen A, the blood group will be A. Antigen is called as protein. So, in a chromosomal pair, 2 chromosomes will be there. So, either AA will be there or AO will be there. AA means codominant, both are equally important. Both are equally important. Or AO will be there, O will be recessive, A will be dominant. If A is dominant, what will be visible? A will. So, whether it can be AA or AO, or simple language, if both are recessive, means what outcome will be there? A only, no? If one is A and other is O, if A is dominant, what outcome will be there? Then phenotype will be what we call it as A. What is that antigen called as? Every time antibody will be against. If antigen A, for that, if antigen A, then B will be there. B will be linked to it. Then it is called as matching antibody. So, antibody will be opposite. B. Remark. A can give blood to whom? You can give to whom? A. Already I have A, so I can give it to A person. Already I have A. AB also I can give. Why? Because in AB what is present? A is present. B blood group. B. Phenotype is called as B. How I will call it as BB, BO. What is antigen called as? B. If antigen is B, then antibody will be A. Then I can give it to whom? B comma AB. Yes or no? I can give to B comma AB. You can give means B can give to. Here you can give means what? B can give to whom? B and AB. AB is there. What is the phenotype? AB. Then what will be the genotype? A and B, both will be present. Here both are codominant, both are equally important. There is no recessive, no dominant. Both are same, A and B. Both are equally important. Then the antigen will be A and B. When antigen A and B both are present, then there will be no antibody. Why? Because A is present. So, antibody B should be there. If B is present, antibody A is present, A should be present. But both A and B are present, then there will be no antibody. So, antibody will be nil. AB, universal. Now, I am what? AB, I have both A and B. Universal recipient means I can take from A, I can take from B. Why? Because I have both AB. Now, last O. O means nil. O means phenotype O. O means nil. Again the genotype is O. When nothing means antigen also will be nil. When antigen nil means antibodies will be A and B. Both antibodies will be. I am what? I have nothing. I have nothing. So, I can give to anyone. Why? Because I do not have any antigen. I have vacuum. I do not have any antigen. So, I can give to A. I can give to B. Why? Because if I come, no one is going to attack me. Why? Because I do not have antigen. If I am O, if I am entering into any body, if I enter into the body called as A, if I enter into the body called as B, if I enter into the body called as AB, if I enter into the body called as O, I do not have any antigen. If I enter, no one will recognize me. Why? Because I do not have. O means what? Antigens is nil. When I do not have antigen, I can go into any body, no. Hence, I will be called as what? Universal donor. I can be present everywhere. Universal donor meaning is what? I can present everywhere. Why I can present everywhere? People cannot recognize me. Why? Because I do not have antigen. So, I will be present everywhere means I am a donor. Here donor means what? I can be present everywhere. When I can be present everywhere and no one is going to stop me means I will be a universal donor. O blood group. That is mainly dependent upon what? Antigens. And this antigen is because of what? A information which is present on the gene. That gene will tell outcome will be antigen. If outcome is A, then the blood group will be A. If the antigen is B, outcome will be B blood group. If antigen is A and B, then the blood group will be AB. If antigen is 0, then the outcome also will be 0. According, there is no need to by heart also. If I am a O blood group, I do not have any antigen. If I do not have any antigen, if I do not have any issues with you, I can come to your home any point of time, you will welcome me any point of time. There way because there is no differences only. But if differences is there at one point of time, 1% difference is there, there is a chance of inhibition. If I go, he might remind about that, why you behave like this, so unnecessarily why I should go. So, there is some inhibition. If there is no issue, then I can go anywhere. I do not have any antigen. When I do not have any antigen, no one is going to fight with me, so I can go anywhere. So, O is called as universal donor, AB is called as universal, are red blood cells, antibodies, antigens, blood types compatibility. Blood group A, what will be the antigen? A. Then what will be the antibody? Antibody, antigen. Then I can give to whom? Blood types compatible in emergency, A and O. A comma O. O can come into A. I can take it. B. B is antigen. B. This is antibody. Antibody A. Then B comma O. AB. A comma B comma AB comma O. AB positive is a universal recipient. Why? Because I have both. So I can receive. I am not giving. I can receive. So I am called as AB is called as universal recipient. O is called as donor. Why? Because I do not have any antigen. When I do not have any antigen, I can go anywhere. When I have both, anyone can come into me. Why? Because I have both. When I have both, I can compatible with anyone. Why? Because I have. So who found for the first time the blood groups? Landsteiner in 1900 discovered that there are antigens or agglutinogens such as A and B on the RBC and 2 antibodies or agglutinins. Agglutinins are antibodies, antigens are called as agglutinogens. Based on the presence of these antigens and antibodies, the 4 blood groups were being found. Yes or no? ABO blood group system is useful when there is blood transfusion. If I can give blood when my blood group is A and your blood group is A, my blood group is A and your blood group is B, I can give blood. If my blood group is O, your blood group is AB also I can give. Why? Because I am a universal donor, you are universal acceptor. This calculation of blood transfusion will be according to blood compatibility. If blood compatibility is not there, then the man might die. So, this studying of blood is very important because of blood transfusion, tissue transplantation. Earlier, tissue transplantation, parentage determination, criminal cases. Earlier what used to happen? Parentage determination, not now, earlier. If there is a problem that son, whether the father is not accepting that he is my biological son, earlier times, it is having some error today, but earlier, if the father is not accepting that he is my biological son, if the blood group of son and the blood group of father both are matching, then they would be conclude that he is the biological father. In that way, the parentage determination used to be done. Third. Fourth, criminal cases. A blood sample was being present on the criminal crime scene. So, if the blood group, not DNA, if blood groups are also matching, they used to come to a conclusion that yes, at least up to some extent. So, we found blood group. From blood group, we can found more things. Or blood is found, blood group I found, from blood group I can go for a DNA fingerprinting, or at least I can check the people who are having this blood group. In this way, the study of blood will be helpful in blood transfusion, tissue transplantation, parentage determination, and ABO system forms an example of multiple allelism. A is allele, B is allele, AB is an allele, O no allele. Means what? ABO blood group system is a best example of multiple allelism. In my gene, chromosome number 9, on the chromosome number 9 there is a gene. Understand this. On the chromosome number 9 there is a gene. The main function of the gene is which blood should be produced. Which blood producing? It can be A, it can be B, it can be A and B, it can be O. So, is it not multiple allelism? It can be A, it can be B, it can be A and B, it can be O. So, there is multiple allelism. ABO blood group system is a best example of multiple allelism. Why? Because blood groups are A, B, AB, allele A blood group, allele B blood group, allele AB blood group, allele O blood group. So, blood group system is an example of genetic polymorphism. Blood group system is also example of multiple alleles. For example, a chromosome number 11 is responsible for skin color. Skin color can be brown, skin color can be black, skin color can be white, skin color can be yellow based upon the gene. So, is it not the example of multiple allelism? Any form I can get. Similarly, ABO blood group system I can get any form. It can be A comma B comma AB comma O. Hence, blood group system is also example of multiple allelism. Blood group system is also example of genetic polymorphism, because one individual will be having one blood group according to the gene, another person will be having his own blood group according to the gene, another person will be having his own blood group according to the gene. Hence, there is multiple polymorphs. Hence, there is genetic polymorphism also. At population level, it is called as genetic polymorphism. At individual level, it is called as multiple allelism. At population level, means when I am seeing from outside the population, it is genetic polymorphism. When I am seeing at individual level, I can see an A, it can be an A, it can be a B, possibilities. It can be A, it can be a B, it can be AB, it can be O. So, multiple possibilities can be found, means what? Multiple. The alleles are A comma B comma O located on the long arm of chromosome number 9. 3 alleles contribute to the phenotype to the blood group called as A, comma AB, comma AB, comma O. It can be A, it can be B, it is a combination of AB, or it is absence of AB. Accordingly, A blood group, accordingly B blood group, accordingly AB blood group, accordingly O blood group. In ABO blood group system, O is recessive, A and B are codominant to each other. The frequency of ABO blood group system is across the world. For example, in the world, people who are having O blood group, the people who are having O blood group are, how many people are there? 47% are there, means they can give blood. 47 means approximately 50% of the population can share their blood. So, O blood group is what? Universal donor. So, approximately 50% of the population can give their A how many? 41. B how many? 9%. AB how many? 3%. Now, you see, this is called as natural selection. I will give an example of natural selection, how nature will select. AB, how many people are there? Only 3% are there. AB, how many are there? Only 3% are there. But nature made them what? Acceptor. You might be a 3%, but you can accept blood from O or A, B. Means who is safer? O is safer or AB is safer? AB. Your people are less, but you are not unsafe. Why? Because you can get blood from O or A or O people are more. O people are more. Why it is called as it is universal donor, but it can get only from O. It can get only from O. You are 3%, but you can get from 100%. You are 47%, seeing 50% people are O blood group, but O blood group can get blood only from. Why? Because if A comes, it will fight. Why? Because A is not there in my body. If B comes, again I will fight. Why? Because B is not there in my body. AB both together came. Then again I started fighting. Why? Because so I can take only if it is coming from O. So 50% population, but limited only to, I can give, I am a donor, but I am not an acceptor. I am an acceptor only for O. AB, I might be only 3%, but I am an acceptor. Who is more safer? O is more safer or AB is more safer? This is called as natural selection. The rare blood group AB, but who is more safer? AB. The rare blood group AB is present in Australian aborigines and Oceania region, Melanesia and Polynesia. The majority O blood group is present in Europe and Asia, population variation. In parentage determination, they found that there are some errors. It is not so authenticate as DNA fingerprinting, but because the baby can also blood group get from mother also. But how you can determine that if father blood group and mother blood group, or father blood group and son blood group or daughter blood group is same, fix that he is an biological father. It is having some errors. The frequency, this is very important. See, this is world. This is world map. This is world map. Now you see, see carefully. The frequency of B blood group is very interesting. The frequency of B blood group decreases and A blood group increases as one proceeds from Pacific coast of Asia towards westwards in the direction of Atlantic coast. You know, this is Pacific, this is Atlantic, yes or no? That is a world map. That is called as Pacific Ocean, this is called as Atlantic Ocean. Now I am moving from Pacific Ocean to Atlantic Ocean, from east to west. Okay, I am moving from east to west, from Pacific Ocean towards Atlantic coast. The frequency of B blood group is decreasing, means frequency of B blood group is decreasing. The frequency of B blood group is decreasing and A blood group is increasing as one proceeds from now you see, when I am moving from Australia towards North America, the people who are having B blood group are slowly decreasing. When I am moving from Australia to North America, the people who are having A blood group are slowly population variation. When I am moving from Australia towards North America, B blood group people are slowly means what? If B blood group are 90% of the people in Australia, by the time I reach India 40%, by the time I reach Africa 30%, by the time I reach Europe 20%, by the time I reach South America it is 10%. Whereas ABO blood group, it is in Australia 5%. As I reached China it is 10%, I reached India 30%, I reached Europe it is 50%, I reached America it is 80%. So, the frequency variation with respect to blood group, as one person moving from Pacific coast of east to the Atlantic coast of west, the B blood group people are and A blood group people are increasing. That is called as frequency variation. That is called as ABO blood group. In the same context, there is another factor which is called as rhesus factor. If protein is present in me, rhesus protein does not have any function. In our body 95% of the proteins are waste, 95% of the DNA is also waste, only 5% is functional, 5% of proteins is functional, remaining all are water. For example, protein, which is necessary. Why? Because it will maintain osmotic balance. So, 95% are waste. In that one waste is rhesus protein. So, in my blood, if rhesus protein is present, it is good or bad? It is present means present only, no? 4 tickets you buy for movie. One friend came and he slept. Again he got up after the movie. Whether ticket you buy or not, he is watching or not, whether functional or not, he is present or not. So, rhesus protein also same. It does not have any function, but if it is present, it is going to impact. Why? Because it is a protein which is nothing but antigen. So, other person's body, if it is enters in other person's body, there will be an antibody. If it identifies as a foreign particle, it will attack. When both will fight, there will be agglutination. When both are fighting, it results in disaster. So, in that way, the rhesus factor is also important, presence and absence. Mainly at the time of birth of a child. If the father is Rh positive, mother is Rh negative and the baby is Rh positive, the first child will not be affected, but by the time the antibodies of the mother will be activated. And if the second child is coming up, these antibodies will go within 3-4 months of the baby's time in the mother's womb, it will go and destroy the baby. That particular disease is called as erythroblastosis fetalis. But however, now we have the medicine, by taking the injection we can neutralize that Rh positive. We can neutralize Rh positive means we can make it unidentified. When it made unidentified, then Rh negative will not attack. No, why? Because it made unidentified. In 1940, Landsteiner and Wiener demonstrated that Rh factor in rhesus monkey is erythrocytes, and they also observed the same Rh factor is also present in human beings, which is independent of blood group. So, whatever may be blood group, rhesus can be present, rhesus cannot be present. Means rhesus is independent of blood group system. Means rhesus does not have any link with blood group system. If A is there, I am a rhesus. I might be present, I might not be. If blood group B, I might be present, I might not. It is independent of blood group system. Means I am telling you, there is no relevance between the blood group system and rhesus. Both are independent. But if rhesus factor is present, then I will call it as positive. If rhesus factor is not present, then I will call it as negative. Simple. The blood group are, we tell O positive, O negative means nothing but O blood group with rhesus factor O positive. O blood group without rhesus factor, then it is called as O negative. We tell no, COVID positive, COVID-19, COVID negative. I know few students don't know this today also. O positive means that COVID virus is present in us, then we are called as COVID positive. If that virus is not there, then they are called as COVID negative. If that rhesus is present, then we are called as race is positive. If the race is not present, then we are called as, in the blood group O positive means O is the blood group and races is present, then it is called as O positive. O negative means O blood group and races is not present, then it is called as, which is independent of blood group. Presence of Rh factor is determined by, again, races is what? Protein. So, protein is because of what? Respective gene. So, anything because of gene it is called as, genetic marker. Hence, races factor is also a genetic marker. Anything because of gene. So, Rh factor, anything because of gene. Blood group is because of gene sitting on the chromosome number 9. Races factor is also because of gene. Hence, races factor is called as, the presence of Rh factor is referred as Rh positive. What I am explaining, again I am theoretically I am telling you, so that there will be a connectivity. While you are reading your PDF, it will become very, very easy. Which contains Rh? Antigen. Antigen is what? A protein. A protein antigen is present, it is called as Rh positive. Absence of Rh factor is referred as Rh negative, which means there is no specific antigen. No specific antigen means no antigen protein, no rhesus protein. When no rhesus protein means Rh negative. On the blood group O, so O negative. On the blood group O, O positive. Rh is a protein which is present on RBC. It will sit on the RBC. Rh system is independent of, I told in the beginning, it is lymphocytes, it is present on erythrocytes. Rh is a protein which is sitting on RBC. It is a dangerous if there is Rh incompatibility. So, only one situation is very, very dangerous, presence of Rh or absence of Rh. That particular situation is called as Rh incompatibility. How, sir? Mother doesn't have rhesus. Mother doesn't have rhesus means mother doesn't have that gene which is responsible for manufacturing of rhesus protein, but father is Rh positive. Both combined together. Mother became pregnant. First child will be normal, which is called as Rh positive. First child will be normal, which is called as Rh positive. But mother is Rh negative only. But by the time I identify there is a foreign antigen, I am mother. In my stomach there is baby. The baby is rhesus. I am Rh negative. Means we both are different. Why? Because I don't have that protein, but baby is having that protein. But the baby is in my body. In my body I doesn't have rhesus. Then my body will identify that Rh positive as outsider or not. So, any outsider I should attack. That is the duty of antibody. It can be a virus, it can be a bacteria, it can be protozoan, it can be fungi, it can be anything. It can be rhesus also, it is an outsider. Why? Because it is not there in my body. So, compulsory I have to attack. By the time I manufacture my weapons and go and fight this rhesus protein, 9 months will be completed. 9 months will be completed. Now, the baby is out, the baby is safe. Second child. Now, again the mother became pregnant. Second child is present in the mother's womb with Rh. If the first child is Rh positive, then second child also will be Rh positive, fixed. Then the child is having Rh positive, but already antibodies are ready. Within 2-3 months I will identify and I will go and destroy. That is called as erythroblastosis futalis. Erythroblastosis futalis. Rh incompatibility. This is the only major function which is associated with rhesus. If not, presence of Rh or absence of Rh doesn't have any relevance on our body. See, population variation. Rh positive. European people are how many? 85% are Rh positive. Rh negative how many? 15%. India 93% are Rh positive. 93. How many percent are Rh negative? Now you tell me. I am telling male and female both together. Europe are Rh positive or 85% and India Rh positive or 90. Where the chance of erythroblastosis futalis is more, very good. Why? Because 80. So 15% are Rh negative. More chance of getting erythroblastosis is in 15%. Who are those? Next, human leukocyte antigen, HLA. Human leukocyte antigen, HLA. What is this human leukocyte antigen? There is a concept called as major histocompatibility. There is a concept called as major histocompatibility. What is this major histocompatibility? Grafting, which is common. That grafting means comparing. Organ transplantation is also called as grafting. My kidneys got destroyed. If I tell someone's kidney got destroyed, they will beat me. My kidneys only got affected. My kidneys are not working. Some people are happy here. So what? I want to exchange kidney. So kidney is a foreign particle. So foreign particle is coming inside me. I am removing my kidney and I am taking some foreign kidney. So my body will not recognize it, obviously. Why? Because it's a foreign kidney. So what will happen? My antibodies will go and destroy that organ. So there cannot be organ transplantation. To have organ transplantation, we need to have major histocompatibility. What is major histocompatibility? Means there is an antigen called as human leukocyte antigen which will be sitting on WBC. When human leukocyte antigen of you and human leukocyte antigen of me, when both are matching, means your antibody and my antibodies will be matching. Antibody is nothing but protein. So I can accept your kidney or you can accept my kidney. So that possibility will be there when there is major histocompatibility. So major histocompatibility is called as comparison between the two human leukocytes antigens. So any grafting, any organ transplantation which is happening on the earth, it can happen only after comparing HLA, human leukocyte antigen. Now we will see. In human beings, major histocompatibility is simply called as HLA. The major function of major histocompatibility, it checks out the quality of being tissue compatible and the ability of donor tissue or organ to be accepted by the recipient. HLA are the human forms of. So generally the concept is called as major histocompatibility. In every organ, in every species, if there is major histocompatibility, then only we can go for organ transplantation. In human beings, what we will be comparing? Human leukocyte antigen. So HLA will, generally the concept is called as major histocompatibility. In human beings, when we are comparing HLAs, it is also called as major histocompatibility. That compatibility will be checked with which protein? Leukocyte. Human leukocyte antigen. These are the proteins located on the surface of WBC, and this HLA is because of the chromosome number 6. So on the chromosome number 6, the gene is present and the gene is responsible for an output called as the gene will be present and the gene is responsible for an output called as human leukocyte antigen. So the gene is responsible for manufacturing human leukocyte antigen, means whether human leukocyte antigen is a genetic marker or not. So first I proved it is genetic marker. Arranging the foreign cells, tissues, organs into the body of a recipient known as grafting or transplantation can happen if and only if HLA matching is happened. HLA is because of result of multiple allelism. Because of HLA, tissue and organ transplantation is becoming difficult for grafting. HLAs of two individuals should be same, then only grafting will happen. Therefore, tissue typing for these antigens is done on the membranes of lymphocytes. Best success has been with tissue typing between siblings and between parents and children. Means there is no community variation, there is individual variation. Sometimes matching will happen, sometimes matching will not happen. Matching mostly happen between the siblings or between the parents and the children. There is no community variation. In the case of HLA, it is matched, it can be used. If it is not matched, it cannot be used. So HLA is nothing but a genetic marker. Because of the chromosome number 6, there is an HLA. If your HLA and my HLA are matching, then only we can go for transplantation or grafting. And this HLA compatibility is most frequent in siblings or in parents and children. The HLA frequency is individual variation but not community. So any transplantation, any grafting which is happening across is mainly because of matching between HLAs and HLA. That concept is called as major histocompatibility. So in major histocompatibility, what we are going to compare? HLAs. So HLA is mainly human being centric. Likewise, if we want to transplant anything, we have to be having major histocompatibility. So that major histocompatibility in human beings is called as, next is haptoglobin haptoglobin. Listen carefully. What is the lifespan of RBC? Lifespan of RBC? 120 days. RBCs are manufactured where? Bone marrow. That is the reason when there is a cancer, they will go for bone marrow transplantation or bone marrow surgery. which is in rarest of the rare cases only it will be successful, if not the person will die, high chance. The doctors will not recommend for bone marrow. Why? Because bone marrow is a sensitive area where that RBCs are getting produced, and every RBC lifespan is 120. So, RBCs, erythrocytes are roaming in my body. After erythrocytes, RBCs are manufactured by what? Hemoglobin. The combination of hemoglobin is called as blood. Hemoglobin means what? Heme plus globin. What is globin? Globin is called as globin class of protein. What it will be carrying? Heme. Heme means called as what? Iron. So, a mineral, iron will be sitting on the chromosome, iron will be sitting on the protein called as globin, and this will be moving here and there. And this hemoglobin will, if hemoglobin is active, we are active. If hemoglobin are less, we are inactive. That is the reason the doctors will tell that you have to improve your hemoglobin, and that increase in hemoglobin mainly depends upon presence of iron. So, after 120 days, RBC, which is manufactured because of hemoglobin, which is nothing but a protein carrying iron. Iron is a mineral which is very costly, which is very useful. After 120 days, RBC will destroy. Why? Because its time period completed, the time period expired. So, after 120 days it will expire. It is a common phenomenon. Why? Because deadline came, 120 days finished. So, that RBC it will destroy. When RBC is destroyed, what is present in it? Hemoglobin is present. In hemoglobin, what is present? Iron is present. Iron is very important, iron is very significant, iron is very costly. Now, this iron is freely flowing in the blood. Why? Because it got bursted off, it came out from hemoglobin, it came from RBC. Now, iron is simply roaming like a drunken person, it is simply roaming here and there. It can fall into the drainage, or it can go into the park and sleep on the bench, or it can destroy anything. Why? Because it is simply roaming. Now, what we have to do? One well-wisher, one friend, someone should come and carry him, put it in auto and take him to the home. Who will do it? Haptoglobin will do it. So, haptoglobin will come, iron please wait, I will carry you. So, it will carry haptoglobin. Haptoglobin, a genetic marker. It will take the iron, it will sit on it, you wait, you control for 5 minutes. It will take up to bone marrow and it will leave it. If not, this iron will go into liver. If not, this iron will go into kidneys, where it will produce what? Bile salts, which results in what? Jaundice. So, jaundice is very dangerous. So, in order to stop this iron, which is very costly, so see, same thing, sometimes just I am telling you, you are very, very important. First, you have to believe that. First, you people should believe that I am very important. I am not waste. I am not suitable. You should not think that you are unsuitable. Every human being, every species, not human being also, every species on the earth is important. Every person is important. Every individual is important. We don't know tomorrow what that person will become. Tomorrow what you will become also no one imagine. Same way, iron is also. Iron is so precious. People will buy it, dates costly iron, they buy it so that the iron will increase. Once burst off, it is freely moving. If it go into the kidney, it will result in jaundice. If it go into the bone marrow, it will produce RBCs. Where you are going is very important, true. On whose company we are living is also very important. Whose company, sometimes God also will not give proper people, but one day He will give us proper people. At that time we have to identify the opportunity and we have to move on. Identifying the opportunity is also a very big thing. We have to identify whether the opportunity is good or not. Iron should identify that yes, haptoglobin came, yes, now to sit on haptoglobin. If not, no, haptoglobin will not come, I am freely roaming here, I am free bird, I am independent. I will, wherever I want I will go, means where it will suck, where the low pressure is there, from high pressure to low pressure. Everything we will move from high pressure area to low pressure area. Where the low pressure is there? Liver, kidneys. So, it will go and fall into the. So, we have to identify what is good, what is bad. Same like haptoglobin. So, it is a protein in the human being encoded by the HP gene. It belongs to the globin class of proteins that binds with free hemoglobin released from the burst of erythrocytes. It helps in iron from RBC under destruction by transporting free hemoglobin to liver. If iron reaches the liver, biliverdin or bile salts will be produced. If produced in excess, it leads to jaundice. Eventually, the damage of liver and kidneys. It is often referred as suicide proteins. Why? Because I myself going and falling into the liver. So, it is called as suicide protein. So, here what? Frequency variation of HP is found. High frequency of HP deficient individuals are found in many tropical African population. In Western Europe, 40% people have HP deficiency and less in frequency in Middle East. Now see, if haptoglobin is present more in me, haptoglobin means what? Globin means protein. Hapto means a hapto variety of globin. If haptoglobin protein is present in me, what is the function? I will not allow the iron to be wasted. I will send this iron to bone marrow. Means useful or not? More the haptoglobin, more healthier or not? More the haptoglobin, lesser the anemia or not? More the haptoglobin, lesser the anemia. Middle East people are having less anemia because haptoglobin is more. Why haptoglobin is more? Because they eat iron rich food like dates. They eat iron rich food like dates. So, however it may be, the iron is pumping into the bloodstream, into the Middle East, Syria, Jordan, Israel, Iraq, Iran, Saudi Arabia, Gulf countries, these all countries. So, haptoglobin is nothing but not allowing the bursted iron to go into the liver and stopping it from going to the liver and sending it to the bone marrow is a major function of haptoglobin. And that haptoglobin is a protein which is manufactured because of a gene called as HP gene. Whether haptoglobin is a protein, haptoglobin is genetic marker, what is transferrin? Same. Now you see 120 days completed. RBCs got destroyed. RBCs got destroyed. Now this iron is moving in the blood, freely moving. Haptoglobin anyway it is doing another duty. Now transferrin. There is another thing which is called as transferrin. What this transferrin will do? This freely flowing iron, it will stop from falling into liver and pump it to bone marrow. But already in bone marrow sufficient iron is present. Sufficient iron is present. So, I will have excess iron. So, when excess iron, that potential will not be there. When excess is there, it will go and fall into the liver. So, excess iron is also a problem. So, what transferrin will do? Transferrin will convert the excess iron and store on the cell membrane. It will catch the excess iron and store in the cell on the outer cell membrane. So that now excess iron which is freely flowing will not fall into the liver or kidney. Why? Because it is excess. Now what I did, this excess iron also I started conserving by converting it. I am storing it on the cell membrane. Whenever one day iron deficiency will be there. So when there is iron deficiency, then this iron I will use and I will pump into the bone marrow. Transferrin is a blood protein which transfer iron to different cells in the body. Whenever there is burst of RBC, the hemoglobin is released into the blood. Macrophage which release iron from hemoglobin into the blood, that particular thing. This free flowing iron is transferred to different body organs and cells like bone marrow and spleen. At bone marrow, RBCs are produced. Hence, transferrin helps in producing RBCs by giving iron to bone marrow. If this will come if and only if there is excess iron. If normal iron, haptoglobin is sufficient. You are weak and your friend is drunken. If he is drunken limited, you are enough. Now you should lift means another friend should come and another friend is transfer. In excess is uncontrollable. So then the next transferrin came. Hence transferrin help in producing RBC by pumping iron to bone marrow. If there is excess of iron, there is a chance that iron escape into liver. Why? Because it is in here. Transferrin acts as a catalyst and stores iron in the form of ferritin. It converts iron into ferritin. Which iron? Excess iron. Excess iron will be converted into ferritin. Ferritin is like a fat. It will be like it will be stored. Ferritin. Ferritin is a universal intercellular protein that stores iron and release it in controlled fashion. In human beings, it acts as a buffer against iron deficiency and iron overload. When iron overload, it converts into ferritin. When iron deficiency, the ferritin will be converted into iron. In that way, in excess of iron, this transferrin will be helpful. Whenever there is iron deficiency, ferritin will release iron. There are 5 transferrin in the human body across different population. This transfer, there are 5 varieties of transferrins are there, 5 varieties of proteins are there, or 5 alleles of proteins are there, which is called as TFA, TFB, TFC, TFD, TFE. TFC is the most common transferrin found in all the populations of the world. TFD is found only in Africa and mostly refers with anemia and Australia. So, that means if TFD transferrin present, I will not work effectively. The TFD transferrin is present where? Australia and in Africa. But what is common? TFC. Normal transferrin is what? TFC, which is present across the population. But TFD is present in where? Africa. That is the reason they are suffering with anemia. African people and Australian aborigines are suffering with anemia because they have a transferrin, but which allele of transferrin? TFD, which will not function effectively. That means what? It is genetical aberration. That is what it is, a genetical aberration. Now, what I will do? I have to make TFD as TFC. How I can do? By doing gene therapy. So, when TFD become TFC, what will happen? The excess iron will not be wasted, the excess iron will be converted into, so individual will not suffer with anemia. Haptoglobin only to pump the bursted iron into the bone marrow is haptoglobin. When iron is excess, then TFB only in Navajo Indians, TFA and TFE are very rare. Classification of 5 transferrins is based on intensity or magnitude and time interval. Delay while reaching bone marrow. With these factors, the effectiveness of anemia can be calculated. That is the reason TFD, where the TFD is there, the people are suffering with